Hello class, my name is Jillian Luthi and I'm presenting my senior research thesis on hot and cold, the effects of imidacloprid pesticide on bumblebees' thermoregulating capabilities. I work with Dr. Sarah Richmond in my mentor's lab on campus, Dr. Ann Leonard. For this experiment, we looked at plant-pollinator interactions. Pollinators include animals such as bats and birds, and insects such as wasps, butterflies, beetles, and bees. While there are many species of bees, such as honeybees, solitary bees, and sweat bees, we focused on the bumblebee. The role of the pollinator in this interaction is to carry pollen on their bodies to transfer genetic material between plants. They pollinate roughly 90% of flowering plants and contribute to billions of dollars worth in crop production and agriculture. To simplify, that means 1 out of every 3 bites of food you take come from pollinators. Unfortunately, the plant-pollinator interaction is in danger due to climate change, habitat loss, invasive species, and pesticides. A specific subset of pesticides we'll be looking at is the neonicotinoid insecticides. The name literally translates to new nicotine-like insecticides because, similar to nicotine, they affect certain receptors in the brain. They are presented as seed dressings or sprays and can be found in soil. They have become increasingly more common since the 1990s, especially with companies like Bayer. While there are multiple examples of this specific type of insecticide, we'll be looking at imidacloprid. Some of the effects they have include decreased reproduction and foraging abilities, neurotoxic effects such as uncoordinated movements and hyperactivity, increased worker mortality, and possibly thermoregulation, which is what we'll be testing. The reason we're testing this is because multiple companies, including Bayer, have claimed that this insecticide does not affect beneficial pollinators. Unfortunately, this has been refuted due to neonicotinoid traces found in pollen and nectar. Unfortunately, this is more dangerous for bumblebees because their queen bees spend more time actively foraging, which means that they can contaminate their larvae, or that means they could prematurely die before their colony life cycle can be completed. This leads us to our research question. Does the consumption of neonicotinoid pesticides affect the bee's ability to individually thermoregulate in changing heat environments? We hypothesize that consuming a neonicotinoid pesticide will keep bees from cooling down in warmer environments and retaining heat in colder environments. This is our timeline. So in September, we established our experiment and research question. In October, we set up our colonies. As seen here, we have our 2 colonies, and we started the trials. We continued those trials into November, where we then finished the month with our data analysis, including the research poster and presentation. So this is a summarized schedule of what the average week looked like during the experimental phase of the research. So on Monday and Wednesdays, you can see the days are identical, and same for Tuesdays and Thursdays. That is because the experiment consisted of 2 parts. So the first part, on Monday and Wednesday, is the setup. So we set up the bees, the dosage, the feeders, and incubate them overnight. And then on Tuesdays and Thursdays were the longer days where we actually ran the trials. So for part 1, which took place on Monday and Wednesday, first we would collect the bees. So as seen here, we have our 2 colonies, and we were taking 20 bees from each colony, so a total of 40 bees. And of those 20 bees from each colony, 10 would go to the treatment group, and the other 10 would go to the control group. To extract the bees, we have this special vacuum that goes in, sucks up all our worker bees, and puts them in a container that we then put in the refrigerator to cool them down, make them sleepy and easier to work with. During that time, we set up our feeders, shown here, and they contain 30% sucrose. Once the bees are sleepier and less active, we then individually place them in these chambers. So the blue stickers represent the control, while the pink represent the pesticide. And they are separated between the 2 colonies. Then they are incubated overnight in a controlled climate. The second part, which usually took place on Tuesdays and Thursdays, is the actual experiment. So first, we remove the feeders, where we once again weigh them, and the bees are starved for 2 hours. This is to guarantee that the bees are going to be hungrier and therefore are going to try to consume the dose more. As for our dosage, we have 20 microliters of 50% sucrose solution for our control group and 20 microliters of 50% sucrose solution plus 20 parts per billion of the imidacloprid. it is 50% sucrose instead of the 30% that they had in their feeder to make it sweeter and more enticing to the bees. And to make that way, we make sure they consume their whole dose. So once the 2 hours are up, we give them their specific dosage depending on their group. And as you can see here, we put the dose in through their holes. We try to look for the more active bees first, so we make sure they actually consume their dose. And once we have seen that they've consumed it all, they are then moved into the next line where they are put into the trial. So once it's the bees' turn for the trial, they're put into this aluminum channel as shown here. And in that channel, they are placed on a heating/cooling plate. So for the heat trial, the plate is heated to 53 degrees Celsius. And for the cold trials, the plate is heated or cooled down to -5 degrees Celsius. And for both trials, the aluminum canal is placed half on the plate and half off the plate and creates roughly like a 10-degree difference inside. And during that time, there's a thermal camera placed up here that's facing downward and records the whole interaction for 10 minutes. Once their trial is up, they are then euthanized in the freezer. As for analysis, so like I said, the thermal camera uses the software called FLIR, F-L-I-R, and it takes a thermal recording, a video recording of the bee during their 10-minute trials. And so once the trials are finished, I went back over and reviewed all the videos, and for each specific bee, I looked at their temperature readings for every single minute for the 10 minutes. Once we have compiled all that data, we used a program called RStudio to analyze it with a linear mixed model and a likelihood ratio test. From there, we created our results. So for both charts, the lighter lines you see behind these darker lines represent each bee's individual reading, while the two darker lines you see in the middle are the averages of all the bees' temperature readings. So first we'll start with the heating experiment. As you can see, the numbers are a little messier and they overlap a lot. That proves that for the heating experiment, the imidacloprid did not have an effect on the bees' ability to cool down in a warmer setting. We believe that is due because in the warmer setting, bees are more active and aware, and so they're able to move around a lot. So during that time, they can move back and forth between the cooler end off the plate and the warmer end that's on the heat plate, thus creating a more sporadic temperature reading. So because of that, the imidacloprid did not have as much of an effect on the bee. However, we then look at our cooling experiment. And here we see a clear distinction between the pesticide and the control group. And in colder settings, as we explained earlier, bees calm down, they get a little sleepier, and they do not move much. To avoid this issue in nature, bees use their muscles to shiver and retain body heat. However, as we explained earlier, one of the side effects of imidacloprid is affecting your neural activity and your muscle movements. And so when you're in this colder setting, the bees need to shiver to warm up, but the pesticide could be restricting that ability to use those muscles to warm up. Thus, they are losing a lot more heat faster than the control group is. So this data proves that the imidacloprid does have an effect on the bee in a colder environment. And here we have our conclusion. As I said, we found that the neonicotinoid insecticide imidacloprid to interfere with the bumblebee's ability to thermoregulate in cold but not hot environments. Future work should prioritize investigating the mechanisms by which thermoregulation in cold temperatures is complicated by neonicotinoid exposure, perhaps by studying its effect on motor function. This study extends the ever-growing body of evidence demonstrating the harms of neonicotinoids to insect pollinators. As we said, that many companies have claimed that neonicotinoid does not affect the beneficial pollinators. However, this study, as well as many others, proves that there is an effect on these bees and many other pollinators. Another reason that this data is so important because although the bees are in danger, bees also contribute a lot, 90% of our agriculture, which means if the bees are in danger, then our crops and our agriculture and our biodiversity are also in danger. Also, The bees are consuming this and there are traces in their pollen and nectar, which means in our food there could also potentially be traces of this insecticide. So it's dangerous not only for bees, but also potentially for humans as well as other animals, even though the effects may not be as devastating on us. And these are all my references, and this is the finalized poster. So while this was an individual experiment, it is part of a larger experiment going on in Dr. Leonard's lab that is looking at the effects of imidacloprid on the bees' regulating capabilities. And it is part of a larger research group that we are looking at through the next year as well. My specific project is completed. I completed the semester. However, I will still hopefully be helping with the overall project we have going on in Dr. Leonard's lab. Thank you so much for your time. If you have any questions, comments, or concerns, please leave them down in the comments and I'll be glad to get back to you. And I thank you for your time. Have a beautiful day and a great end of semester. Thank you again.